It's my pleasure to introduce Maitreya Dunham. Maitreya got her PhD in genetics at Stanford and was a Lewis Sigler Fellow at Princeton before joining the Department of Genome Sciences at the University of Washington, where she recently became a full professor. And today, she does a variety of work on yeast genetics and evolution, but here we're going to hear specifically about her work on beer yeast. So take it away.
Great, thank you. Uh-oh. Okay, I'm back to being co-host, sorry. There we go. Okay, can you see my slides? All right, and I'm gonna bring the chat up so that if anyone has questions throughout my talk, you can absolutely feel free to stop me. Stop and interrupt, ask questions. If I'm in the middle of something, then I'll see your chat message and I'll answer it once I get to a good break point. But it's always fun to put together a talk for a broad audience. And occasionally I hit too high or too low in terms of background knowledge. So please let me know if you don't understand a term that I use or if you think I could explain something a little more thoroughly. So I'm really excited to talk with you all tonight about my lab's work on watching evolution in real time happen in the brewery. And this, I think, is a really interesting topic because one really fascinating area of genetics is understanding mutants, understanding how have all of the organisms that humankind has had some interaction with and domesticated for our own use over millennia, how has that actually occurred? How do we have, a wide variety of different plants and companion animals and agricultural animals that have different traits that we have been selecting them for all sorts of useful things. And while the organisms shown here are things that might come to mind immediately when you think about domestication, this little microorganism, Saccharomyces cerevisiae, the budding yeast, is actually also an organism that has been domesticated by humans. And yeast has, of course, been used for all sorts of different fermented beverages over history. I'm just showing a few of them here, but it's also, of course, used for bread fermentation. It's involved in coffee and chocolate fermentation, among other things. So how people have been using yeast for our own devices over time is just, in my opinion, a fascinating topic. And yeast also happens to be just a great system to work with because it's not only been domesticated for use in, say, fermented beverages, but also for use in the laboratory. It's a really common system that we use to understand genetics and biochemistry and cell biology. And that long history of work in the lab has made it a really easy to study organism and one that we can manipulate and actually test hypotheses that we come up with about how domestication works. So one thing that we can really appreciate about yeast and its history, thanks to modern tools of genome sequencing, many strains have now been sequenced from all over the world and from all sorts of different niches. And what I'm showing here is like a big family tree of yeast. So you can imagine all these little lines that connect the yeast as kind of who's most closely related to each other. And so these groups here represent different styles of beer that are being made by different types of yeast, which are closely related to one another. And there's just been a ton of activity now of sequencing all these yeasts and trying to understand their genomes. However, one thing that makes this really difficult is that two different yeast strains can have thousands and thousands of polymorphisms different from each other. If you compare their DNA sequence, they can actually be really different. And so that makes it really hard to say, well, what does make a wheat beer yeast different than a lager beer? Well, actually, lager is an easy one because lager turns out to be a hybrid between two different species. But for these ale yeasts, which are all Saccharomyces cerevisiae, what makes an American strain different from one of these other strains? And it's unfortunately really hard to just stare at their genomes and try to figure this out. There have been some really good advances along these lines where people have been able to pick out some genes that are responsible for certain metabolites or certain behaviors. But overall, it's been a difficult problem. And so one thing that my lab has started to do is ask, well, what if yeast aren't done being domesticated? What if they are still evolving and we could capture that and watch it in real time? So instead of this situation where we have all the tips of this tree and we don't have these ancestors that are kind of historical in nature, what if instead we could move evolution forward and watch how it happens? What if we could say, here's a mutation that occurs and starts to take over a fermentation and see what that mutation is and how it affects the yeast and its behavior and even phenotypes like how it makes the beer taste. And it turns out that this is a good idea because it's exactly what brewers do. So brewers actually reuse their yeast over and over again and are kind of doing evolution experiments all the time in breweries all over the world. And so the idea here is that you buy some yeast from a yeast supplier, or sometimes you have your own frozen stock that you can set up, and you inoculate a big fermenter with it. And then once that brew of beer is done, you can take some of that leftover yeast and use it to start the next fermenter. Do it all over again, start a new fermenter, start a new fermenter, start a new fermenter. And in kind of modern breweries, the number of times that you undergo this cycle is on the order of 5 to 8 repitches before you stop. And so, a good question is, well, why would you stop? Why not just keep going? And so this is a great cartoon that I found on a brewing blog that is kind of like the brewer's understanding of evolution, where, the more times you reuse the yeast, the more likely you're eventually going to create zombie yeast or something. So what is zombie yeast? Well, this is kind of a catch-all for, the yeast start misbehaving, they start dying, they might get contaminated, they might stop creating the flavors that you want. And so, you don't want to make beer with zombie yeast because, that might not be a product that you want to get behind. So many brewers stop before this point, but we were interested in finding a brewery that would kind of go out for a longer term, kind of historically how brewers must have treated their yeast, and see if we could find a place where we could in fact do the experiment and watch evolution as it happened. And so luckily I got to meet Tom Schmidlin. Tom runs a brewery called Postdoc Brewing. And Tom, as you might imagine from the name of his brewery, is trained as a scientist. And so he has been really just fantastic to work with and was really game to kind of brave the zombie threat and see how far we could push the repitching and see what we could find in the yeast genomes. And he also makes some delicious beer. If you haven't tried his beer, for those of you in the Seattle area, this is my favorite one because it's both a yeast pun and a delicious IPA. Alpha Factor is a mating pheromone that yeast use, and that's actually what's drawn on the label here. And so, buying this at Whole Foods cracks me up every single time. And so Tom was able to go out to 26 cycles for us on our first experiment together, and kind of we were off to the races. So the second component that I need, in addition to a long time course where we can give the yeast a chance to evolve, is a way to watch evolution. How are we going to, discover those different lineages and those different mutants that might or might not be happening as the yeast are being reused? And that's where some modern tools come in that we use in my lab to study genome evolution and in other, lab controlled contexts. But we realized that we could use the same tools to understand how yeast are evolving out in the world in this this different environment. And so a modern genome sequencing lets us sequence every base pair of these genomes for a reasonably low cost, such that we were able to collect time courses, and you can actually follow, if this red strain here has a single point mutation in its genome, one single base out of the 12 million bases in the yeast genome that changes, we can actually watch that spread over time and know where it is and what gene it's in. And again, because yeast has a lot done in the lab about it, we can know what that gene does in many cases and kind of extrapolate from there about what it might have to do with how it's behaving in these fermenters. Okay, so the experiment. We're going to take one of these time courses. We're going to sample as frequently as we can, and then we're going to use genome sequencing to see if the genomes change. And if they do change, then we'll try to understand how they change and what that has to do with domestication. Okay, so the first time course that we did with Tom and Postdoc Brewing, he had purchased this yeast, and this is an American strain of yeast. It's part of a big family of yeast called the Chico varieties, and that's because they were popularized by Sierra Nevada Brewing, which is in Chico, California. And so this strain is a great all-purpose strain. And so Tom actually uses it for most of the different beers that he makes, with the exception of certain ones that demand particular flavors. So usually what he does is he buys the yeast and then expands it so that he has enough for fermentation. And then he starts off with his first beer. And in this case, that was a pale ale. And then he made a porter. And then he made another pale ale. And then he made 3 batches of IPAs. So this is pretty typical, where you can take that yeast from one batch of beer, and sometimes you get enough back that you can split it across multiple batches. And so there's some forks in the road here too, where he made both a blonde and a wheat wine from the yeast that he harvested from these IPAs, and on and on. So this is the real life of a brewery, right? So this took months and months and months. It's one of the longest experiments that we've ever done. And we're really thankful to Tom and his crew for harvesting the yeast for us and bringing them by the lab. Luckily, one of his team members lived pretty close to the University of Washington, and so was able to drop off yeast samples for us on his way home. And so what we did was we found the kind of linear track through all this. So we didn't go down those branches, but we went just from one beer to another. And this gave us 26 repitches. And then we didn't sequence all of them. At first, we were like not sure what we were going to find, right? So we instead chose 5 of the samples to sequence. So we wanted to see, what did we start with? What did we end with? And can we see some of the action in between, in terms of like, could we see things rising or decreasing or what? And I should say that Tom decides to stop his repitching when he's just not happy with how it's behaving. There's either like an off flavor, or the yeast are not behaving as well. And so we left it up to him how long these experiments should go, and for the the satisfaction of what his product is that he's trying to develop here. Okay, so we sequenced all these samples, and before I tell you the result, I first have to introduce you to the ale yeast genome. And that's because the ale yeast genome is pretty amazing and pretty complicated too. It's been really great fun to work on. So historically, the origin of the ale strains is likely to have been from a mating event between an Asian wine strain, like something that might make sake or another related kind of beverage, and a European wine strain. And each of these strains already had genetic differences in its genome. So you can think of this as, the wine strain here got 1 chromosome from yeast mom and 1 chromosome from yeast dad, and those were different from each other. And the same here for the Asian strain. And so what happened here is this resulting strain actually has 4 copies of every chromosome. So unlike us, where we have 2 copies of every chromosome, this yeast actually has, you can imagine it as like 4 different parents, right? So so all 4 of these copies of the chromosomes all have the same genes, but they're all a little different from each other. They have different alleles of these genes. So then what happened? So this is the kind of initiating event. And then over hundreds of years of brewing, these strains have diversified. So, you can get different chromosomes that behave in different ways. And so we can see some chromosomes where, we go from having 4 copies to only having 3 copies. And other chromosomes where we keep 4 copies, but the parent of origin from each of those copies changes. And so that was what our question was. So when we look at these strains now, and compare all the different brewing strains that are out there, they're really different from each other. We can see whole chromosome copies that are at different numbers. We can see recombination events that swap up arms of chromosomes. And that's what makes it really difficult to understand which of these are relevant for changing the traits of the yeast. And so what we were hoping is that if we propagated it in the brewery, that we would be able to find a smaller number, and that we would be able to link those more directly to the traits. I got a great question in the chat about what informed the choice of which pitches we selected for the intermediate time points. And to be honest, we just spaced them out pretty evenly, because we weren't sure in this first experiment how slow or fast were things going to happen. And so one nice thing is, because we essentially have the entire fossil record of this brewery time course in our freezer, right? So if we found out that we wanted more time points between 2, spaced apart time points, we could go back into the freezer, get more out, and sequence them. So it kind of was our starting span set, and then we could always go back and do more. Okay, so I'm going to show you a little bit of data. So what happened? Well, what happened is the chromosomes changed even more. So I told you that some of the changes that are different from all the different strains are things like extra copies of chromosomes or missing copies of chromosomes. And this is one case where we saw that happen. So this is chromosome 5. This particular strain, the ancestor starts out with 3 copies of it. And then by the end of the 26 repitches, we ended up with 40% of the population had 4 copies of this chromosome. So that's a huge amount that has had this mutation happen. So what's happened here is we had probably a small number of events of this mutation, and then those were able to grow better or survive better or have some other beneficial trait and increase in their proportion of the population over time. So don't think of this as this mutation happened independently in 40% of the cells in the giant fermenter. That would be a really unlikely. So this is instead an example probably of mutation and then selection. And the way we know that we have an extra copy here is by we just ask how many sequencing reads. When we sequence the genome, we split it up into a bunch of little pieces, and then we can count how many times we sequence each region of the genome. And so in this case, we start out with sequencing each region of the genome. This is like over the whole chromosome, just little bins, and we get an average copy number of 3. But by the end here, we see an increase. And if we play that out into the probabilities, then we end up with about 40% of the population has an extra copy. The other type of mutation that we saw, so this is another chromosome change, but it doesn't actually change the copy number. Instead, it changes essentially which parent those chromosomes are from. So here's an example just with people. So, you have one chromosome from your mom and one chromosome from your dad. You can have mistakes, though. There are mistakes when cells divide, where instead of getting one chromosome from mom and one from dad in the next generation, so you have 2 cells that are identical to each other like you should. Instead, you can have, say, both of the green chromosomes go to one cell and both of the blue chromosomes go to the other cell. So this is an error in chromosome segregation. And this sometimes happens in human genetics even, where a child can have their chromosomes from one parent and not the other, or a cancer cell can lose a copy of a chromosome from one origin and make extra copies of the other one. And the reason that this can matter is, let's imagine that a gene on this chromosome has a non-functional copy on the blue chromosome, but a functional copy on the green chromosome. So in the situation here, where you have one copy of each, usually this functional copy is enough to provide whatever activity this gene is providing. However, this situation here where you have this unbalanced chromosome segregation, this one's going to be okay. It's got functional copies of this gene, but now this one here, this daughter cell, even though it still has 2 copies of this chromosome, both of those copies carry the non-functional version of that gene. And so this could be a case where, whatever that gene is required to do, that might be a problem and cause a disease or cause a trait that is undesirable, or in some cases cause an advantage. So sometimes in cancer cells, for example, mutations that cause the cells to grow better or to stop breaks on the cells are playing out with some patterns like this. Okay, so what did we see in our yeast cells? We saw the same kind of thing. And this is a little bit of a complicated graph here, but this is a chromosome that already has some of these chromosome rearrangements in its past history. So instead of 4 parents that we're observing now in the modern strain, this chromosome arm, this side here, has 3 of the parents left, and one of those is at 2 copies. And then this side of the chromosome here has only 2 of the parents left. One of them is in 3 copies and one of them is in 1 copy. And the way that we know that is by looking at different nucleotide bases in the DNA and asking what frequency do we see them in. And so this side, let's start with this side here because it's a little easier. So in this case, we have 3 things that are one type and 1 thing that's another type. So when we look at our frequencies here, we find 1 out of 4 and 3 out of 4 are the major frequencies that these arise at. So this side of the chromosome here, we have some 50-50s, and we have some 3-to-1s, and we have some 1-to-3s. So you get kind of a variety of things here. And so what we saw was this chromosome that already started out with this rearrangement actually became even more rearranged over the course of our repitching time series, where you can see that it started out at 3 quarters and 1 quarter, but by the end, they're kind of like coming more close to about 50-50. And so when we went and we looked at individual clones to confirm what was going on, what we found was that these were strains which had converted one arm of a chromosome to one of the subparents. And so they're back to this 50-50 arrangement instead of this 3-to-1 arrangement. And you might be thinking, well, the way you've cartooned it out, like, why are you getting these kind of like, slopes here? Why isn't it a step function? And we were initially a little confused about that too, until we started going and looking at clones. And when we sequenced a whole bunch of individuals, what we found was that this was a mutation that hadn't just happened once. This had been a mutation that had actually happened multiple times in the population. And then all those different mutants had kind of together started growing more in the population. And so we are seeing, some that have a large swath of that chromosome and others which are just a teeny bit at the end. And so all of them share this bit at the end, which is why it's closest to 50-50, but only some of them go this far into the chromosome. Okay, so that is our one time course that we did with Tom, which took a long time. But I'm a scientist. I like replication, right? We were amazed that we saw all these changes over just this short period of time, relatively speaking, for the natural history of yeast. And we wanted to know, would we see the same mutations again if we were to redo the experiment? And luckily, Tom's got a very productive brewery and was game to start over again and collect samples again. This time, he went for 29 repitches. And to our great astonishment, we got the same mutations again. So when this happens in a genetics experiment, it's really lucky, because in many cases, there are multiple ways that a yeast strain could adapt to a new environment. But what this told us was that these mutations really must be doing something to help the yeast survive in this environment in order for us to see it in 2 completely separate runs. But both of these were in Tom's brewery. Maybe Tom's doing something specific that is leading to these changes being selected for. And so we were really lucky to be able to collaborate with 3 additional breweries, including Elysian, Drake's, and Red Circle. And I have to thank Escarpment for setting up one of these collaborations. And these strains are all related to each other, these starting strains, but they have some things a little bit different. And that's one thing that we noticed, was that this strain, for example, already had one of this extra copy of chromosome 5 that we were getting over our time course in these other experiments. So what we think we're doing here is actually recapitulating some of the mutations that have already been happening over domestication of how these strains have been propagated historically. But now when we're paying attention over a small time course, we're being able to see some of the same processes and even some of the same mutations. And so we were really just amazed that we were able to see, some of these same mutations occur at a totally different brewery using different recipes, different styles, and different starting strains that are related but slightly different from each other. Okay, so what do these mutations do? Right? So, one thing that is interesting about these chromosome scale mutations is it's often unclear what individual genes are involved in creating the traits that they have. And so Chris Large, who I should have mentioned at the beginning of the talk, sorry about that, Chris. Chris Large is the graduate student who did all of the genomics work for this project, and also made most of the slides that I'm showing tonight. And Chris has a real eagle eye. And so he was poring over these genome sequences one day, and he realized that one of the genes changed more than others. So it has a point mutation, a single base pair change in it, and it's on one of the majority chromosomes here. But all of our rearrangements that make 2 copies of this dark blue region replaced that region. So you'd think that they would have 3 choices to replace the dark blue with the light blue, or the light blue with the dark blue. But in fact, in every time we looked, what we saw was that this copy with this particular mutation in it was the one that was being purged. And so that seemed very suspicious. And so when we looked at the identity of that gene, it was a really interesting one. So this is a gene called BAT1. And what BAT1 is it's a gene that encodes an enzyme that is involved in metabolism of amino acids. So it actually sits in your mitochondria. Humans have an equivalent of BAT1 as well. And it sits in the mitochondria and it converts a precursor into a valine, and also can do the same reaction to create leucine and isoleucine. So these are all amino acids that are used for core metabolism in yeast and growing and making proteins. And also this feeds into other parts of metabolism as well. So you can see how some of the other molecules that are linked in this network are all these other alcohols. And so some of these alcohols are things that are undesirable in beer. They create kind of a solventy taste. And so Chris and I were really, really lucky to go to a conference where we met what turned out to be the world expert on BAT1, Professor Hiro Takagi. And he and his team volunteered to study this mutation with us and try to understand what consequence it was having on yeast metabolism. And so I want to thank them for their collaboration with us. And what we were able to do was they recreated this mutation in not in a beer strain, but in a lab strain. And so that just made it easier to study and easier to tell differences of what the mutation was doing. And so what they did was they made these strains with different genotypes, different mutations in them, and then they tested the levels of some of these different alcohols that are kind of things that are created by this metabolic pathway. And so a normal sequence that is the normal strain sequence has low concentrations of these alcohols. If you delete the gene entirely, you just knock it out of the genome and completely mutate it so that it's gone, then you see a higher level of these. And when you make the beer strain sequence, this abnormal allele here, we see that it looked very similar to what happened when you deleted the gene entirely. So to us, this implied that this is a broken allele of this gene, and our evolved strains, our strains that we find at the end of our repitching, are actually getting rid of this bad mutation. And in the process, they might be creating less of these alcohols. And in fact, we did do some experiments. Tom brewed beer with 2 of our evolved strains and our ancestor. And I got to bring these to a home brewing conference where we did a blind taste test, or excuse me, sensory analysis is what they like to call it, among all of the participants. And we were actually able to detect differences in flavors among the strains, the beer that they made. We also, in my lab, created what we fondly call science beer. Noah Hansen, who's a technician in my lab and who actually got interested in microbiology through home brewing, he's created a really reproducible wort recipe that we can use in the lab to make beer in the most reproducible way that we've come up with yet anyway. And we were able to send some of that off for analysis of the chemical content. And we were able to detect differences, not just in the levels of these alcohols, but also in some other flavor molecules, like you might have heard of diacetyls. That's what causes that kind of like buttery popcorn flavor. And some people are really sensitive to that, and really a little bit of diacetyl goes a long way. Some of it adds some nice mouthfeel too, but it's a balance, right? And so depending on the strain, it had more or less of some of these flavor molecules. But you might be thinking, what does having a different concentration of these other alcohols have to do with your ability to grow and survive over months of propagation in the brewery? Like, why would these be beneficial? And so it turns out that another phenotype, another trait that this mutation is relevant for is the ability to tolerate high levels of glucose. And so what this is showing is this is the gene deleted entirely, this is the normal copy, and this is the beer strain copy. And what this is showing is yeast diluted a little bit at a time until you get down to single colonies. So this is like a patch of yeast with a bunch of yeast there. And then each one of these single colonies started from a single cell. And so it gives you a way of comparing their growth in a really easy to do way. And so you can see that at low percentage of glucose, this is our normal glucose concentration that we normally grow yeast at. These are all growing pretty similarly, but as the glucose concentration ramps up, you can see that the normal allele, the strain with the normal allele, grows better than the strain with the gene knocked out, or the strain carrying the beer allele. And wort starts out with a pretty high sugar content. So we think that this could be one reason why this strain has both a flavor phenotype and also a growth phenotype that could lead to its changed proportion over time in the fermenter. Okay, I'm going to talk about one more gene. And this was a really interesting one. So this is a gene that is on that same chromosome region. So just downstream from BAT1, we've got another gene called FLO1. And FLO1 also is really interesting to us. And I should say, we actually had to resequence our strains in order to discover this. We collaborated with Joseph Cheshire's lab to use a different sequencing method that lets us get, instead of little snippets of DNA like the Illumina sequencing does, this lets us get long regions, but those regions have more errors in them, but they help us figure out what genes are next to each other. And that was how we were able to figure out that one of the copies of the chromosome has this FLO gene on it. And so it's linked to that, that other flavor gene. And this is a gene that we got really interested in because it's related to a trait called flocculation. And flocculation is something that yeast does, and it's something that brewers are really interested in because it is really helpful in the brewing process. So in the lab, the yeast strain that we use has this knocked out. We actually, years ago, it was discovered that the most popular lab strain has a mutation in it that makes sure that the yeast will not do this. And that's because if we're doing microbiology, we want the cells to grow all mixed up and all free floating. So when these are budding yeast, remember, so when they bud off this new daughter cell, we want that daughter cell to float away. And so that's why this tube here, which we've just mixed up, is kind of opaque. So if we shined light through it, we would not be able to see anything on the other side, which is how we measure the density of these cultures. However, this is a strain which has this capability. It's able to flocculate. And so what happens is, instead of these daughter cells going away, the cells are able to actually stick to each other, and they have special proteins on the surface of their cell, which is kind of like molecular Velcro that causes them to stick together. And so instead of free floating cells, you end up with clumps. And you can actually see these clumps with your eye, right? Like, these are teensy cells. They're only like 10 microns. And yet these are, macro clumps that you can see really well. And so one way that we measure this is we mix up the culture really well, and then we let it sit for a while, and we measure how long does it take for it all to sink to the bottom. And so you might imagine that this is a really useful trait if you're growing, instead of just a little tiny test tube of yeast, if you're growing like a big fermenter as tall as like a 2-story building, right? Like, how are you going to get the yeast out of your beer at the end of your fermentation? Well, if it flocculates and all sinks to the bottom, then that's very convenient. So flocculation is a trait that brewers care a lot about. Okay, so just to demonstrate that, this is another way that this mutation could be beneficial. This isn't causing the yeast to grow better. It's causing them, let's say we have one strain here that's now got this mutation that has more of this flocculation. And now it can sink to the bottom better. And because the harvest is usually done from the bottom here, then it has a higher probability of being taken to the next round. So this is a good reminder that, these ideas about who's the best mutant, there are lots of ways to be a beneficial mutant, right? You can grow better, you can survive better, you can be more likely to be taken by the brewer into the next cycle. There's lots of different dimensions to what could cause a particular mutant to be successful in a brewery environment. Okay, so did our mutants have this phenotype? So what Chris did was he used the same test tube assay where we put some of the strain in a test tube and then we let it settle, and then he takes just the top part. And so if you go back to these test tubes here, you can imagine if we just take this top part here, this will be cloudy and this will be clear. And so we can just use a gadget in the lab to measure that. Let's see how much light is allowed to go through that culture. And so here are the controls. Here are our starting strains, just 2 replicates of that. And then here is a positive control that we knew would flocculate. So that has 100% flocculation in 6 minutes. So our mutants here, that have, they either have that extra chromosome that I talked about, we're actually still trying to figure out exactly what's so great about having this extra chromosome. This one is a little bit more of a mystery than the rearrangement, actually. So still lots to figure out here. And then we have two other clones that don't have anything on this chromosome that has the recombination. They do have other changes in their genome, but they're not common ones in the whole population. And you can see that these look pretty similar to our ancestor here. And they also have gotten rid of this BAT1 mutation, I should mention. So this is why we don't think BAT1 is relevant for this particular phenotype. But when we get these recombination ones, we see that these they have different parts of the chromosome that have been swapped from the light blue to the dark blue, but they all have an increased flocculation. So what we think is going on is that we're seeing an increased amount of this flocculation here. Okay, so why in the world would this be happening now? So this is a question I often get. It's like, well, if yeast have been adapted for hundreds of years for brewing environments, then how much more could they possibly evolve? And so this is a case, I think, where what we're finding is a difference in how brewing is done has changed over even just the last 100 years. So this is a photograph that Chris found in a textbook of a brewery called Ballantine Brewing. Ballantine actually is probably the source of the ancestor of all these Chico strains that we've been working on. And instead of waiting for all the yeast to settle out to the bottom and then use those to start that next pitch, instead they use a practice called top skimming. And so this is where you have the yeast foam rise up to the top, and then you're able to scoop off the yeast foam and use that to go forward. And so this is actually historically a really common process. And so our hypothesis here is that, with the advent of easy freezers that you could archive strains and freeze them down and save them indefinitely, that we kind of have halted evolution of these strains. And that now once you order them up from a yeast company or grow them up yourself for creating your beer, that they're now being put in a different environment than what they were historically grown under. And so we might be seeing that now they have to adapt to this new environment versus what they were experiencing at the time that they were archived. So I think this has a lot of fun implications. One, it means that yeast are still adapting. We are able to actually see yeast growing and being domesticated for different environments that are, as we create different features, we see different characters of these yeast, and different traits are beneficial for them. In this really interesting genome region that we saw recombine, we saw that this flavor gene, which also has a growth phenotype at high glucose concentrations, is linked to a flocculation gene. And so this is kind of an interesting model for how traits could co-evolve. It means that you get one mutation, but you get multiple traits occurring simultaneously. And so this could be one possible explanation for, how do you do some sort of selection on flavor if that flavor change doesn't actually change anything about how the yeast grows? It becomes really difficult. You can't, screen millions of different beers and pick the one you want. I mean, you can over a long period of time, but it's a difficult system to imagine that working. So this is a case where having all those genes co-localized could create a kind of a whole cohort that goes together. I've talked about mutants and how they can be both good or bad. So, they're good for the yeast, right? These are creating more intense flocculation probably. There are, but is the flavor that they make good or bad? Who knows, right? That's up to the personal tastes of who's drinking it. And so when we made our mutant beer to compare to our ancestor beer, Tom actually liked one of the mutants better. So you can imagine now that maybe Tom or another brewer would use this evolved strain now as the new house strain. So it's kind of been pre-adapted now to his particular brewery conditions. And now the next time you make a beer with it, it doesn't have those mutations to explore because they're already there. And maybe we'll start getting different mutations, but it might be a way of avoiding certain paths that happen over and over again. We have put some of this information in much more detail in a manuscript that Chris wrote, and with our contributions from our collaborators. And you can find that manuscript on a website called bioRxiv. So if you haven't run across bioRxiv before, it's a place that people put manuscripts that aren't peer reviewed yet. So buyer beware, you do need to keep that in mind, that this is things put up with very little filtering. And one way that it's being used by scientists is to get information out there in a quicker way. So, we're also going to publish this in a peer-reviewed journal, and we're working on that. But in the meantime, you can read our results as we interpret them on this preprint. And I want to end with just a few caveats, right? So first one is that we haven't actually, I kind of misled you. We weren't actually looking at every single gene in the genome, and that's because of a quirk with how we do the sequencing analysis. The easiest way to do sequence analysis is to compare everything to a beautifully, perfectly sequenced reference strain. And in yeast, we've had the genome sequence since like the mid-90s. And so our reference sequence is just gorgeous. However, it's of a lab strain. And that lab strain doesn't have certain genes that are important for brewing strains. For example, it doesn't have the genes that are related to maltose metabolism, which is kind of shocking when you think about it, a yeast strain that can't eat maltose, right? Also, like I mentioned, we don't want our lab yeast to flocculate. And so it's gotten, we've gotten rid of some of those genes as well. And so analyzing these genes is requiring special analysis. And I'm lucky to have a couple of great undergrads on the project who are targeting these genes and trying to understand them specifically. The second caveat is all the trait information that I showed you, besides the BAT1 mutations, which were reconstructed in a lab strain, we haven't reconstructed those mutations in a brewing strain. And we really would like to do that in order to be completely sure about causation. These strains often also will have a smattering of single base pair changes throughout their genome, or other mutations that we're really not sure yet how they contribute to the differences in how these strains behave. And so Chris has been experimenting with using CRISPR technology in order to engineer some of these strains to have some of these mutations, and only those mutations, so that we can do more careful comparisons. And then finally, the experiments that I showed, which I think Chris calculated represent something like 4 years total of beer. It's a lot of work by the brewers to put all this together. But they were all using these related strains. And so we're really, really interested to understand, how does the strain background matter for the capability of that strain to evolve more when it's now put back into the brewery. And so we're looking for additional collaborators who are using other strains. I've got one collaborator who's doing top cropping on a strain called London 3, and I'm really excited to see how that ends up. We've been trying to find collaborators using lager yeast, which, like I mentioned, is a hybrid between Saccharomyces cerevisiae and a whole other species called Saccharomyces eubayanus. And then I would love to get repitching time courses from other products. I went to a brewing conference last month and learned about all these breweries who've started making hard seltzers. And it is truly astonishing the amount of sugar that goes into those to get the fermentation going. And I would love to see what is happening to the yeast that are now being used for those. And there is a lot of really interesting research being done by these breweries about what are the best strains, how do you treat them, how can you keep them healthy. And so being able to capture things like that, ciders is another one that I'd love to get more samples of. So I have been really happy to be introduced to new brewers through people who've heard my talks, or I've gone to talk at homebrew clubs and been able to meet some brewers that way as well. So if you are at all interested or know somebody who might be, then please let me know. And so here's my contact information here. I'm Maitreya at uw.edu. I'm also on Twitter if you want to hear the latest and greatest about not just beer yeast, but other things that my lab is up to. And I want to end by just thanking all the great folks in my lab. So here's Chris wearing his Postdoc Brewing shirt. And here's Anna and here's Noah, who are also part of the team. And I want to thank our great collaborators, and also all the people at the breweries that were kind enough to work with us and to let me go out in public and say there are mutants in their beer, and to help us understand some of these mutations and traits as well, and give us ideas. And then finally, this project was started as a part of an NSF grant to understand how hybrids behave and evolve. And it was funded as part of what's called the broader impacts. NSF is always really interested in making sure that scientists are interacting with more people than just the folks in our lab, that we're thinking about problems of the world and that we're interacting with other people besides just other scientists. And so this was originally funded as a part of a broader impact for one of these grants, but it's ended up being just so much more and really, really exciting and exciting to work on. So I've been really happy with that. And the American Society for Brewing Chemists actually just recently funded our continuation of this project. Okay, so with that, I'm going to stop sharing my screen so that I can see you all, and then I will answer some of the questions that are cropping up. All right.
Thanks so much, Maitreya. You seem to have full command of the chat, so I'll just let you carry on with that until people raise their hands.
Yeah, great. And you can feel free to unmute yourself and ask your question, or I'll just go through the chat and call on people. So Stephen and Kara, do you want to ask your questions out loud, or do you just want me to read them? I will just read them then. Would you expect to see a higher degree of enrichment of BAT1 corrected alleles after brewing a higher gravity beer? Or is that difficult to determine sensitively enough over a single round of selection? That is a really great question, because especially in a brewery like Tom's, where he's using the same strain for so many different styles of beer, you might imagine that one that starts out at a lower sugar concentration would not have as strong of selection as one that starts out with a lot of sugar. And I bet that's true. We only have the end point of each one of these. So I think that you're right that it would be difficult to really capture. But what we could do is we could make wort in the lab with different recipes that only differ by their sugar amounts, and see if we can recreate that growth difference in wort in the lab. So I think that's a good experiment idea. Thank you. Sai. How come the zombie yeast effect isn't as much of an issue for sourdough starters that are kept going for years? Is this because of the bacterial relationship? Another great question. So this is my white whale. I would love to get an experiment like this going for sourdough starters. And I wanted to do it in the lab because I wanted to control everything. And so the thing that I'm actually stuck on is sterilizing the flour. I have not been able, there's all sorts of horrible fungal spores in grocery store flour, which is fine, they're not actually horrible, but they contaminate all my experiments that I've tried to do this. And so I would love to get a time course. And there are actually some people out there working on sourdough starters and getting really, really interesting data about like how the microbiome on the baker's skin interacts with what ends up in the sourdough starter and things like that. I think that there are almost certainly adaptations of the sourdough starter strains to that environment, because it's really another strong, narrow selection. It's like, you imagine a yeast out in the wild and it's probably eating a variety of things, but you're giving it only sugar and flour forever, right? So, it's really going to have to become a specialist on that. And so I wouldn't be surprised if there are differences. And some baking strains also have really remarkable, complicated genomes, although others are a little simpler. So we're actually thinking about how we could do some genetics on some of these ale strains, and baking strains are on our list because they might actually be a little bit more amenable to that. Yeah, but I just love the idea of baking a loaf of bread with the ancestor strain and the evolved strains and seeing like if you can tell a difference and stuff like that. But yeah, I don't have a good answer for that one. Celeste, do similar evolutionary processes occur during wine production? Another one. Yeah, so we think they likely do, but that idea of repitching is much less common for wine, and that's because wine is very seasonal, right? So you're unlikely to be doing a bunch of serial batches of wine that you would take the yeast into the next one. So in general, wine is not made in the same practices, but it is likely the case that the strains that have been used for wine, they certainly have a variety of genome changes that are really characteristic of that group. And so we did a long time ago with a collaborator some evolution experiments with some wine strains to try to come up with a cold tolerant strain. And so there's definitely room for the wine yeast to evolve as well. using some of these kind of same concepts.
I was thinking in terms of in the wine itself, how the glucose levels might change over time. So not from season to season or pitch to pitch, but just that glucose tolerance test that you're
absolutely. No, and the strains do have to have a pretty high tolerance to high sugar concentrations, and there's also different kind of nutrient balance of the grape must, which is like they care a little bit more about nitrogen, and there are some other things that arise in some of the other molecules from the grapes, which can be inhibitory to yeast growth, and so there's kind of some differences in what they're faced with that they almost certainly are adapting to. And you also have the other feature of all the other microbes and organisms that have come in with the grapes. And so often you set up like a big competition of where the yeast are trying to outcompete all these contaminants, and it's a kind of a race too. And so the yeast that are frequently used for inoculating wines are really good at what they do, but there are phenomena such as like a stuck fermentation where it doesn't go as fast as you should, and sometimes there are special strains that people will add to kind of try to goose things, and other some strains are better at that first stage than at that last stage. So there's definitely some variety there already, and different strains are used for like champagne yeasts and stuff like that. You might remember there was like a big fad of brut IPAs a while ago, of like beers made with different yeast that are more frequently used for wines and stuff like that. So there's a lot of cross-pollination as well from fermenters of all types. Okay, question from Clay Wright. Is it known how flocculation is involved in top cropping? I would imagine to some extent it is necessary to capture CO2 bubbles. I think you're probably right about that. There are some really interesting complexities to flocculation behavior. Jerry Fink, who's a famous yeast geneticist, I heard him talk a few years ago, and he said that there are some Japanese brewing conglomerates that actually own patents on some of the flow genes because they are so important and have different features from each other. So, and also the responsiveness to things like oxygen concentration and ethanol concentration. Like you can have different genotypes that will behave differently depending on the nutrient environment as well. And so that's all really complicated. We were working on one of these genes, Flow8, a few years ago, and it's the control region of that gene is huge. It's like way bigger than the control region of most of the other genes that we studied, and it ended up causing a bunch of practical problems for us when we were trying to clone it onto a plasmid. But there are all sorts of inputs to regulate these genes and how they behave. And so, but I think you're right that they create kind of these rafts that that go up to the top. The other thing that is another related but different trait is in sherry. Sherry makes this floor that can go to the top, and that also is related to flocculation, but in a different way than what we see in the beer brewing system. Another one, can flocculation in yeast is comparable to the origin of multicellularity? All cells in flocculating yeast have similar transcriptome or more heterogeneous? Yeah, that's a really interesting question. You do see, like I said, here's this teensy microbe, but now you can see a clump that is like a multicellular object. And so is there any kind of division of labor or behavior that is similar to how a multicellular organism behaves? And so there's definitely some evidence that cells of like genotype can kind of recognize each other better and are more likely to stick together. And also that the cells on the outside of the flock are exposed to an environment and exposed to potential stressors that might protect the cells on the inside. But at the cost of those cells on the inside not necessarily having access to nutrients or oxygen as much as the cells on the outside. So there are certainly differences in physiology, even if everybody's genetically identical in one of these flocks. A second type of yeast behavior that creates clumps is where the mother-daughter cell division that never is complete. And so that daughter cell, instead of floating away or instead of just sticking, actually stays attached by where that bud neck is. And so that means that all of the cells in that clump are all genetically related to each other. Whereas in a flock, you can get cells that are from different cell divisions that aren't related to each other. They can all stick together because they have all the same cell cell wall markers. So there are differences in how selection acts if everybody's a clone versus if things are genetically mixed up. And there's a couple of different labs that are working on that as a trait. Will Ratcliffe and Michael Trevisiano are using that as kind of a model of multicellularity. And at UW, Ben Kerr has actually done some work with them on some really interesting like modeling to try to understand some of the things that they see in their model of multicellular yeast. But it's really a fun trait to work on. So they don't just do these clumps in liquid. They'll also, if you grow them on an agar plate, they'll make patterns. And so I knew we were onto something when my grad student Elise started working on this trait, and she would have people stopping by her bench to visit her yeast and look at the pretty patterns that they were making. And so we're still working on this, some related traits to some degree, because they are kind of more charismatic than your boring smooth yeast colony that we're mostly used to. All right.
So I'll ask
that
a
covers it for what's in
question.
chat. So sorry, Kelly, I talked over you.
Oh, I was just going to say, since the chat questions are all over, I can ask mine, which was,
Go for it.
so you said that it was sort of up to Tom to stop the repitching when like the yeast got suspect by his estimation. Did you ever sequence like the very last batch to try and identify mutations that caused it to go bad by like whatever definition he was operating under?
You know, we were really interested to see if we'd see something, you know, just so I think it's partially a threshold effect too. And also like when we went back and made new beer with that was all with clones. So we isolated a single cell with one genotype and then made beer with that. Whereas the beer that he made the decision about stopping was actually a mixture of mutants. So it was like 40% of this mutant and 40% of this other mutant, and then some other things thrown in. And so we're kind of interested in how that whole you know, population might work together to create traits that no single clone is able to do on its own. So we didn't find any kind of smoking gun for what might have been what he tasted. And, you know, he's got a great palate. And so he often it's been really fun occasionally getting to do tastings with him, because he'll notice something. I'll be like, oh yeah, all right, let me try that again and see if I can notice it now that you've pointed it out. So, you know, he often will stop things before it's even detectable by like a customer. So it's kind of a, and also it's not just related to flavor, it's also related to like the viability and if it's like active and doing well and stuff like that. So Adam has a question. Is there any potential to collaborate with brewers to genetically engineer superior yeast strains? If so, what traits might be most desirable? Really interesting question. So there's actually a company called Berkeley Brewing that they have engineered the terpene biosynthesis pathway into yeast to make what they're calling self-hopping yeast. So it makes some of those flavors that hops adds to beer, but the yeast is making those flavors. And so that's, of course, a genetically modified organism, which has certain pros and cons in terms of customer acceptance. But I think that their argument is partially that, you know, certain like hops growing is, you know, a consumer of all sorts of resources. And so if you could, you know, offset some of that with the yeast strain, maybe that would be better as a kind of environmentally friendly beer. It's unclear to me if I haven't heard from them if they're commercially using it for things, but I think there are other traits that people have been talking about engineering or even just doing mutant screens for. And so, you know, there's all these esters that yeast make that change flavors. You know, you can, like I said, you can modulate that flocculation behavior. I'm really interested in how things mix together and in like open fermented beers and sour beers, where you have not just Cerevisiae, but other yeasts and bacteria as well, and how those interact. And could you engineer communities that create different types of traits? We have done some sequencing of open fermented beer. We actually discovered a new species in one of them. So they're really complex. So that's one reason we kind of backed off of messing with the communities for a while. But I think there's lots of possibilities there. The other application that we got approached about a few years ago actually was better yeasts for low alcohol beers. So how can you make a beer taste like all the pleasant things about a beer, but with less of the alcohol, which is in demand right now in the marketplace? And so, could you imagine targeting certain genes that don't mess up everything else in metabolism? That's one thing that is always good to keep in mind is, I showed that little snapshot of one little section of metabolism and how a mutation here could affect a metabolite elsewhere in the network. And so you really have to watch out when you go and start engineering these things, because you could create a strain that, accidentally makes a bunch of acetate, and, you end up with vinegar beer and stuff. So it's all connected and really complicated. Carol, you have your hand up. Carol, you can unmute and ask your question if you want.
Thank you. Yes, I've both a sort of question / comment, and then a real question. I've noticed that it's much easier to explain to people what I do for a living in the case of malaria and evolution of resistance, now that everybody's thinking about it so often in terms of COVID. Has that happened to you as well?
Well, it is very compelling to watch evolution in action. It's true. Yeah. I think that the place where we've seen most of an effect, it's really interesting talking to brewers. Brewers already kind of think about this a little bit and are a little already worried that, they call it drift, where the strains change, which is not how we talk about drift, but in any case, we have a high school teaching lab where we're having high school students do evolution experiments. And I think there have definitely been questions coming up more frequently and how those students are relating to those experiments. So I do think that, understanding, just having like genetic trees enter the mainstream as a way of understanding sequence evolution has been fascinating to watch. I feel bad being so scientifically interested in it when this, this terrible pandemic, but I do think that it is connecting people to evolution in a really tangible way.
Yeah. Now, which is a slight benefit of all this horrible other stuff. The real question is, have you ever had a chance to interact with the people who are doing things like Guinness? Very different kinds of, it's not even beer, it's ale, I suppose, or whatever the right word is.
Yeah. I think that, so one thing that I have had a difficult time identifying is, breweries that do the same beer over and over and over again, all in one row, because that's uncommon for most craft breweries anyway. So we have a few leads with some of the larger breweries that do a little bit more of that. And in particular, some of the places that make lager, they'll only make one style of lager because there's not quite as much diversity in lager styles yet in the marketplace. And so I think that I would love to find breweries that do all stouts and then see what that looks like compared to our, mixed one. So if any of you know the head brewer at Guinness, let me know. and as long as they don't mind that I'm going to tell them their beer is full of mutants, Yeah. I have been really excited to see how interested professional brewers are in this. They're interested practically in that they're like, okay, yep, maybe I'll stop at about 15. That sounds pretty good. But they're also interested in some of these mutants and what they do and how the strains are different from each other. And one thing that we found is that strains that are related to each other and even have very similar names actually have some genetic differences from each other. And so, things that people assume are the same are not really the same. And so there's been some real interest in, what can we add to understanding the beer family tree, right?
Yeah. No, this is fascinating. I love your talk.
Thanks, Carol. All right, one last question from Raghu. Could you do analogous experiments starting with wild yeast strains to look at the early stages of domestication, or is that an impossible experiment? Why, what an interesting idea. So I have a collaboration actually with a group in Argentina. They are the first people to isolate Saccharomyces eubayanus, which is the other part of the lager yeast hybrid. And I've had beer that they made and Heineken actually made a special beer for the conference that we were at called Wild Lager. And let's just say a little goes a long way because it is not domesticated. It still has a gene that makes one of these metabolites that it creates that kind of a clove flavor, which is, you often get in certain beers on purpose, right? But you drink one of them maybe and be like, okay, we had a whole beer crawl where it was all eubionospheres. And yeah, by the end of it, we were like, okay, we can get our last stamp, we can do it. But they have been working with local brewers where repitching is not as common. And so they're starting to work with people doing more repitching. And they also are much bigger consumers of dry packets of yeast versus the liquid the liquid pitches, which are more common in the US. And so we're trying to figure out how to get their samples from Argentina to here is where we are on that project right now. But I'm really hopeful that we can get some of those samples and kind of watch some of the more wild ancestor, how it adapts to these environments. So I think you could do it. The early stages just might not be so great, but that's another case actually where engineering, which was Adam's question, can really come into play. So there are several groups that have done things like knock out that gene. There's 2 genes that are required for that kind of off flavor that people don't like. And so if you just knock them out, then you're done, right? So then you can start with that strain and evolve things. And people have been doing that and also making new hybrids and evolving them to see if they can like recreate the early stages of lager yeast domestication. So I think those are all really interesting really interesting ideas. All right, one last one. Can I take one more?
I know you'd said it was last one, but if you want to go back
another one cropped up. So,
means.
Are liquid yeast packs for homebrewing clonal populations, or is there some heterogeneity there? Tangentially related, since yeast need to be expanded prior to the initial pitching, would you expect there to be any expansion of particular clones prior to the selection pressure of brewing? Thank you for asking. You're almost a plant, because we did see some stuff already from the starter cultures. We had one case where the starter culture was already 10% mutant. So there is clearly a lot of selection already happening from the early propping stage. And that makes a lot of sense, because you're going from a very small number of cells to a very large number of cells, and that's exponential growth. And so let's say you've got a mutation that occurs early in the growth. That's going to lead to a large number of cells in the final amount. And also the yeast companies often will make like a very large amount of yeast and then divide it up for all their customers, right? So there is definitely heterogeneity in the liquid packs. The home brewing ones, I haven't looked as much at that. We've mainly sequenced from the commercial ones that our brewers get, and that's both from yeast companies and also from internal yeast propagation systems. So. And that expansion, I think you're right. That during that expansion, there's selection there too, and often that expansion is done in a kind of enriched wort, right? So there's definitely chances there for there to be similar selection conditions, in addition to maybe some different ones that. A lot of times there's like a molasses base that's used for some of these things and stuff like that. So there's different sugar content sometimes. So it could be that the mutants that are beneficial there might not do as well in the beer, or vice versa. So it's definitely something that I've talked to some of the yeast prop people about, and they're thinking it's been fun to learn about their process. So. All right, I think that polished it off. So thank you all for coming. I really do appreciate it.